Hello students, how are you people? Hope you're all doing good, studying very well. So, once again welcome back to our biology class. Myself Dr. Ravindra here. In the earlier class, we discussed variety of biomolecules such as we discussed about how basically the proteins made up of, they are basically made up of amino acids, and amino acids are linked to one another by the peptide bond. And then how the carbohydrates are made up of, carbohydrates are basically made up of the sugars. So they can be trioses, tetroses, pentoses or hexoses also. How the lipids are made up of? Lipids are made up of the glycerol and fatty acids. Well, we have discussed about the simple lipids also as well as the complex lipid such as what we call it as mainly phospholipid we have studied. Fine. So the structural units of these biomolecules very essential before we get into the details of the physiological role of these biomolecules. Well, what is remaining is we need to discuss the structural units of what we can say nucleic acids. So let us understand how these nucleic acids are mainly becoming so lengthy chains or the polymers. Yes, we are going to study that. So structural units of nucleic acids. Structural units of nucleic acids. Nucleic acids, when we talk, yes, they are, we can say that DNA as well as RNA. Deoxyribonucleic acid. So the sugar is deoxyribose. At second position, there is only H, not OH group. Ribose sugar, yes, it is C5H10O5. Why? Because at second position also it is having the OH group. So oxygen is retained. So it is the ribose sugar. Well, so when we talk about mainly the nucleic acids, whether the DNA or RNA, yes, they are basically made up of, they are the long chain of what we can say, yes, nucleotides. So then what is the nucleotide is made up of? Yes, nucleotide is made up of mainly nitrogen bases, sugars as well as the phosphate group we can say. So sugar phosphate forms the backbone of the DNA or the RNA. Well, let us understand the details as such. So when we talk about mainly the nucleic acid as such, consider the DNA itself, right? So DNA, so now the DNA is like this as such. It is mainly, so here it is a 5 dash and here it is a 3 dash we say, and here it is a 3 dash and this is the 5 dash. Why they are denoted as a 3 dash and 5 dash ends, that also we are going to study now. So generally here we can say that adenine always pair with the thymine and guanine always pair with the cytosine. Well, so this, yes, the basic information I hope you might have been studied at your school level also. Adenine always pair with the thymine, guanine always pair with the cytosine. So when we say that adenine A and guanine, what we say it is mainly, yes, G. So these are called as purines, and purines are mainly, yes, purines are heterocyclic compounds. Heterocyclic. Okay, the purines heterocyclic compounds, what is that actually? So they do have what we can say mainly the heterocyclic structure like this. So here mainly they have the heterocyclic structure as such. So the position 1, 2, 3, 4, 5 and 6. So 7, 8 as well as 9. At first position they have nitrogen, at third position they have nitrogen, at seventh position also they have the nitrogen, and ninth position also they have the nitrogen. So the adenine guanine are the purines, they are heterocyclic compounds, they have the nitrogen at, we can say that first, third, seventh and ninth position. Well, so this is about mainly the purines. Okay. So the other thing, what we can say that mainly, yes, the thymine, cytosine. Thymine is denoted as a T, cytosine denoted as a C, and one more is there in the RNA. It is called as uracil. Uracil is U. So these are called as pyrimidines. Pyrimidines. These pyrimidines are monocyclic compounds. Monocyclic compounds. That means they have only the single ring structure. Okay, the single ring structure, and yes, they have the first, second, third, fourth, fifth, and sixth carbon, but at mainly the first position they have nitrogen, at third position they have the nitrogen. Well, so mainly we can say that thus they are called as nitrogen bases only because of having the nitrogen in their structure. So they have nitrogen at the first and third position. Well, so this is all about the pyrimidines. Okay, purines and pyrimidines are the most important structural units of what we can say mainly the DNA as well as the RNA. Now, looking into how actually they are arranged and why these 3' and 5' ends are denoted, let us study that also. Well, so I hope you got a clarity. Adenine as well as the guanine, they are called as mainly purines, and thymine, cytosine, uracil, they are called as pyrimidines. Pyrimidines are monocyclic compounds, and then purines are heterocyclic compounds. Well, so their arrangement, let us understand now. So basically when we talk about yes, so base pairs as such, adenine, these are nitrogen base. Okay, pair with mainly it is pairing with the thymine. Yes, and now the thymine, yes, attached to a sugar. Okay, attached to the sugar. So here it's the terminal oxygen, we can say. Here also adenine also attached to a sugar. Okay. So then further, similarly like this, guanine is a nitrogen base, and then it is attached to the mainly cytosine by 3 hydrogen bonds. Okay, 3 hydrogen bonds. Well, so now this is also again attached to the sugar. So here this is what we are discussing all about the arrangement of the nitrogen bases. So here also it is attached to the sugar. Well, so here the sugar has the first carbon, second carbon, third carbon, fourth carbon, here the CH2, the fifth carbon. Okay, similarly, yes, here is the again OH group will be there. And now when I talk about yes, this is also, yes, we can say that here also the first, second, third, fourth, and yes, CH2, the fifth carbon. Similarly, here the first, second, third, fourth, and fifth carbon, yes, CH2 is the fifth carbon. Similarly, here also the first, second, third, fourth, and fifth carbon, yes, it is the fifth carbon, CH2 is here. So what happens always, this fifth carbon and the third carbon, they are attached by the phosphate group. And similarly here also this third carbon and the fifth carbon attached by the phosphate group. Means here we are observing one end is 5', here it is a 3'. Means the fifth carbon to third carbon. Means this is one base pair. Here in the left side, fifth carbon to third carbon attached, so it is a 5 dash to 3 dash. Here also fifth carbon to third carbon they are attached, so this side is called as the 5 dash to 3 dash. So that's why it is always denoted as the 5 dash to 3 dash. So when I talk about a nitrogen base, yes, what actually the bonds? So these are all what we can say hydrogen bonds. Hydrogen bonds. And then these are mainly glycosidic bonds. Glycosidic bonds. And then what is this? This is what we say phosphodiester bond. Phosphodiester bond. Okay, this is what we talk about. The three important types of bonds that can be seen in the DNA. Yes, they are phosphodiester bond, they are glycosidic bond. Phosphodiester bond between the sugars of the one after the other. Okay, one to the next successive sugar. Yes, it has to attach. So fifth carbon of the one to the next third carbon of the next sugar. So it is a 5 dash to 3 dash. So that side also same thing. 5 dash fifth carbon go and attach with the third carbon having the phosphodiester bond between them. Well, so here glycosidic bond, a nitrogen base and a sugar is attached by glycosidic bond. And then the nitrogen bases, always the purine attached with the pyrimidine, means adenine always pair with thymine, guanine always pair with the cytosine. So the bond between the adenine thymine is 2 hydrogen bonds. The bonds between guanine cytosine is 3 hydrogen bonds. So these are the different types of bonds that can be seen in between the, yes, mainly the bases as well as how the DNA is made up of. Now when we talk about basically a nitrogen base and a sugar, we can say that nitrogen base, so it is called as a nucleoside, means what? Nitrogen base plus sugar, it is called the nucleoside. So there is one more important thing, what it is. So it is called as nucleotide. Nucleotide is what? Nucleotide is a nitrogen base plus sugar plus phosphate group. Phosphate group. This is what we talk all about mainly. How actually the nucleotide now. So nucleoside is only nitrogen base plus sugar. What sugar we have? It is a deoxyribose sugar in the DNA. So this is the structural details of the DNA. What DNA is made up of? It is made up of deoxyribose sugar, phosphate group, and or we call it the orthophosphate. And it is also made up of mainly the nitrogen bases. These are the structural units of the DNA. And yes, nucleoside and nucleotide. So they can be denoted as nucleosides. Let me give you what are the nucleosides. How they are named? It can be adenine, guanine, third cytosine, fourth thymine, fifth in RNA it is uracil. So when we talk about nucleotides can be, yes, it is adenylate acid, yes, guanilic acid, third cytidilic acid, okay, fourth thymidilic acid, okay, fifth uridylic acid. Yes, so thus what we talk about mainly, yes, the DNA is made up of these many important components or the structural units of the DNA what we find. So this is all what we say the structural units of the nucleic acids as such. So we discussed the structural units of the lipids, we discussed the structural units of the carbohydrates, we discussed the structural units of the proteins, we discussed the structural units of the DNA or RNA also we have discussed. So what is required? Yes, mainly to first basically we need to understand how these biomolecules are made up of, what kind of a bond is there between them. Yes, we have studied now. Now the next thing we need to understand the biomolecules and their physiological importance. So we will be understanding the biomolecules such as mainly the proteins, then the carbohydrates, their physiological importance, and nucleic acids and their physiological importance. Yes, we are going to study. Well, so I hope now this point is very clear. What is that? The point is here what you are looking, mainly the DNA. DNA is a double helix structure which is basically made up of the purines and pyrimidines. So the nitrogen bases adenine guanine are the purine, okay, and thymine cytosine, these are the pyrimidines, and then always it is a spiral structure, always the strands mainly run opposite to one another. So this side it move to this and other side, yes, it moved like this. So anti-parallel to one another. So they are attached by 2 hydrogen and 3 hydrogen bonds. Adenine, thymine 2 hydrogen, guanine, cytosine 3 hydrogen bonds. And then when talk about adenine, guanine, yes, they are the purine heterocyclic compounds having nitrogen at first, third, and seventh and ninth position. Whereas thymine, cytosine, uracil are mainly the pyrimidines and they are having mainly the monocyclic compound. They are having nitrogen at first and third position. And then as such the details when we look into this, mainly how they are attached to one another. They are attached, yes, mainly by the hydrogen bond, glycosidic bond and phosphodiester bond. So what is this actually when we talk about the bond structures? Yes, we are finding 2 hydrogen bond between adenine, thymine, 3 hydrogen bond between guanine, cytosine. So a nitrogen base always attached with the sugar by glycosidic bond, and sugars of one after the other attached by phosphodiester bond. So only nucleosides you are talking. They are nucleoside means nitrogen plus sugar. So nitrogen base plus sugar, it can be adenine, guanine, cytosine, thymine and uracil. If a phosphate group also attached to them, then what they are called as adenyllic acid, guanylic acid, cyclic acid, they are thymidyllic acid, uridylic acid, they are the nucleotides. So these are, yes, millions of nucleotides join with one another and they form the polymer, and thus the DNA or RNA is the polymer of nucleotides. So one important thing you need to remember, nucleic acids are the polymer of nucleotides. So here only let us mention DNA or RNA, they are the polymers of nucleotides. So this is all the thing. Yes, I hope you got the clarity about this now. So let us go ahead the next concept as such. So we need to understand mainly what we can say the physiological importance of these biomolecules. So let us go ahead with the physiological importance of the biomolecules. The first we will discuss about basically the proteins. So proteins are made up of what? Proteins are made up of amino acids. Well, so when we talk about the proteins, lots of things are there to understand in the protein, because in every cell thousands of proteins we can find, and proteins can be enzymes, proteins can be hormones, proteins can be receptors. Okay, so proteins are found in a number of ways. Yes, maintenance of the homeostasis, we can say, regulation of body temperature, okay, or metabolism, construction of the tissues, okay, transportation of the substance, in number of ways. So biomolecules and their physiological importance. Physiological importance we are going to discuss. So firstly we will discuss about the proteins. Proteins as such, they are the polymers of, we can say that amino acids. So they are the polymers of amino acids, and then so proteins are very essential for the body. So proteins, the physiological, they form, we can say that they are the receptors. Okay, they are, we can say that hormones. They can be enzymes. Okay, they can be the antibodies. Well, so they play a vital role in the body metabolism. Proteins play vital role in, mainly as such, what we can say that they play vital role in protection, okay, metabolism. They give the protection, they perform the metabolism, they also help in homeostasis. Okay, yes, they are important for tissue construction. They are essential for tissue repair or tissue damage. Tissue repair, essential for tissue repair, and even essential in transportation of substances. Okay, so thus in various way the proteins are helpful for the human body. Okay, so not only the human, entirely we can say as such. So in the nature we can find variety of proteins, and they are basically very important also. So the type of proteins, and then they do have important functions, but in the nature abundant protein, in the nature or in the biosphere, the abundant protein is mainly rubisco. What is this rubisco? It is a ribulose bisphosphate carboxylase oxygenase. Basically it is found in the metabolism of the plants. Okay, rubisco, ribulose bisphosphate carboxylase, is an enzyme that is very much abundant in the entire biosphere. Then abundant protein in the animal kingdom. Abundant protein in animal kingdom. In the animal kingdom, what is that? Animal kingdom, it is a collagen. These you need to remember, because collagen forms what we can say the ground substance. So integral protein, yes, between the different kind of tissues we can find. So there are number of other proteins with lots of physiological importance of the functions. So let us make a list of that. What are they? The proteins and their functions as such. So here when we look at the protein, basically the collagen. Okay, yes. So collagen, then there is a GLUT4, then there is what we can say that insulin. Okay, then there is a trypsin. Okay, then there is antibodies. Okay. Okay, and then there is what we can say mainly, yes, collagen, GLUT4, insulin, trypsin, as well as one more important protein. Yes, first let us understand collagen forms the ground substance. Okay, GLUT4, what we can say that glucose transportation. Okay, insulin, yes, it is an hormone. Trypsin is an enzyme. Yes, antibodies fight the infectious agents. Okay, so these are most important or abundant proteins as such, what we can find mainly in the human body, as well as yes, most importantly we also should remember there are certain receptor proteins. Receptors, yes, on the cell membrane, yes, mainly responses to the stimuli, responses to various stimuli. It may be the smell, okay, it may be the what we can say that the chemicals, okay, it may be the smell or it may be the chemicals, it may be the taste, number of things, yes, we can say that. So thus the collagen is a ground substance. Yes, you might have been studied mainly the structural organization in the animals. There we can find that collagen fibers. Okay, so collagen fibers are very essential in the mainly what we can say that the tissues such as the cartilaginous tissues, so connective tissues. Yes, we can find the presence of the collagen and it is a ground substance, we can say. Then GLUT4 is mainly the enzyme, and it is important in facilitating the transportation of the glucose in the liver as well as the muscles. So excess of the glucose, yes, it remains in the blood. You may suffer with the diabetes. And then insulin is a hormone produced by the beta cells of what you can say pancreas, and it is also involved in glucose homeostasis. So glucose homeostasis. So it is also, yes, called as the hypoglycemic hormone. Means it reduces the excess glucose is there, means in the blood it reduces and mobilizes the transportation of the glucose towards the muscles as well as the liver. Trypsin, yes, it is an enzyme that disintegrate the tissues or the cells, especially helps in digestion also. Antibodies, we call them as the immunoglobulins, they fight against a variety of disease causing organisms. Receptors also are proteins that found on the cell membrane. They mainly respond to the what you can say particular kind of hormones and triggers further any kind of metabolism. So this is all what we are going to study in detail. Yes, I hope you got a clarity what actually the mainly the proteins are. Their proteins are the polymers of amino acids. As such, amino acids are attached with one another mainly by what we can say by the peptide bond. So we are going to understand how actually the peptide bond and what actually the peptide bond formation. Yes, we are going to study the primary, secondary, tertiary and quaternary structure of the proteins also we need to understand. So this is about what we have studied: collagen, GLUT4, insulin, trypsin, and then antibodies. Antibodies also called as immunoglobulins. Immunoglobulins. Yes, immunoglobulins are 5 types: IgG, IgA, IgM, IgE and IgD. 5 type of immunoglobulins are there. So receptors are mainly, yes, membrane and cytosolic, membrane bound membrane bound and cytosolic, we call it as. So these are the types of what we can say the receptors we can find. So receptors are essential for the trigger various kind of biochemical reactions. Well, this is all about for the today's class, and yes, in the next class we will discuss some more essential and important concepts. Till then, take care, study well. Thank you very much.